Chiral resolution kits by ChiroSolve define scalable chiral separation method within matter of days. Background: chiral molecules are closely related mirror image molecules with positive or negative optical rotation, very often with different bioactivity. Worldwide single enantiomer drug market is over $300 billion, 40% of the total drug market. 9 out of 10 top-selling drugs are chiral in nature. Number of chiral molecules as new drug candidates entering the preclinical stage per year are 400, while the number of chiral drug molecules being considered as potential candidates are 40,000. Going from 40,000 to 400 with minimum concentrated research expenditure is a major challenge. Methods of development. There are three categories of technologies available for chiral method development: traditional, asymmetric, and biological. Traditional technologies have been around for a while the asymmetric and biological are newer technologies. Although asymmetric synthesis is gaining adoption, method development is complicated and lengthy process, while the biological methods are still in early research phase. Among traditional methods, chromatography is popular but is not scalable to manufacturing scale. Diastereomeric crystallization used by ChiroSolve is the most scalable and widely used chiral separation method. In fact, 65% of commercially available chiral pure products are manufactured using diastereomeric crystallization method. Here is the typical diastereomeric crystallization example. The racemate ibuprofen is mixed with resolving agent minus phenethylamine and solvent 2-propanol. Mixture is heated until homogeneous and then cooled. One of the enantiomer, negative isomer in this case, crystallizes out while the other isomer stays in the filtrate. Separating out the crystals and treating them with strong acid yields the enriched isomer, which then can be further purified through multiple recrystallization until the target purity is achieved. Diastereomeric crystallization challenges: identifying the optimal separation using diastereomeric crystallization involves choosing the right reagent and solvent out of hundreds of possible combinations. Doing this work manually is monotonous, tedious, time-consuming, and error-prone. Typically, scientists pre-select small sets of reagents due to time and resource constraint and choose the best combination out of these. This bias pre-selection leads to sub-optimized method development. Availability of starting material at this point is a major challenge during early research as well. The solution: ChiroSolve products. There are two products. The screen kits offer more than 380 commercially viable separation conditions to screen the racemate against and shortlist the ideal separation conditions that can be explored further during method development. In Ansho prep kits, they include the screen kits. Additional components are provided to recover the racemate after screening. And additional components are also provided to perform repeated purification until target enantiomeric purity is achieved. It's ideal for medchem and early process chem projects to quickly get small quantities of pure enantiomer, and it identifies how many recrystallization steps are required to achieve the target purity. This separation method is scalable to kilo quantities. ChiroSolve advantage. Operational advantages include consistent results, full exploration done within the kits without handling individual vials eliminates the human errors. Exhaustive screening offers unbiased results and increases the project success. Shortlisted optimum conditions provide alternate routes if needed and help expand the IP coverage. 384 parallel experiments offer tremendous ROI, considerable time, money, and resource saving. Enansho Prep offers full method development and delivers highly pure isomers within matter of days. Strategic advantages: Minimizes the research costs and risks by finding the optimum strategy early on. Standardized approach to method development through easy to follow prescribed procedure eliminates any procedural errors. Unbiased results ensure the full exploration of separation methods. Here are some of the success stories. First example, the discovery group from Big Pharma was unable to separate out the enantiomers, but using ChiroSolve kits, they were able to identify 4 combinations of resolving agents and solvents that gave good results. As a result, they were able to take their project to preclinical stage. Second example, an early stage startup company needed very quick turnaround time to explore different separation methods for IP prosecution. Using ChiroSolve kits, they were able to quickly do exhaustive exploration to find all separation conditions leading to patentable results. Third example, a process chemistry group from major pharma used the ChiroSolve kits to get higher purity. But while doing so, they found a better condition that reduced the recrystallization steps from 4 to 3. As you can imagine, eliminating one recrystallization step from manufacturing can save millions of dollars. Fourth example, a CRO firm had multiple route possibilities for their intermediate and could not decide which one to use. Using the kits, they were able to explore both the multiple routes and choose the best derivatization route that would give them optimum end results in terms of enantiomeric purity and yield. ChiroSolve screen kits offer 384 combinations of resolving agents and solvents, 4 kits per racemate, each with 96 vials. Each kit contains 8 resolving agents and 12 solvents. You get very quick results. Method development is done within a matter of days. Vials and racks are chemically inert and can withstand extreme temperature. As a result, whole experiment can be done inside the rack without taking out the vials. This eliminates the human errors. No additional setup is required. Kits are ready to use straight out of the box. Screen kits features. They're designed to screen any type of racemate. Racemate can be acid, base, alcohol, aldehyde, ketone, or amino acid and can have single or multiple chiral centers. Exhaustive screening provides most separation conditions and quick results. Resolving agents and solvents are chosen with literature precedence. We have selected resolving agents that have historically given good results. Compliance and traceability. Only federally compliant reagents are used. We do not use any controlled substances. Kits design and usage procedures allow easy traceability. Reagents are easily and cheaply available in the market. So when you define the separation process, you get an economically scalable method. Kits are conveniently designed for easy robotic manipulations for high throughput screening. Kits are AutoStation compatible and come with peelable seals to accommodate direct injection of racemate by liquid dispensers. Types of screening kits. They come in 2 formats. Depending on the amount of racemate available, you can choose which one to use. Kits with plastic vials need 12 millimole of racemate. They allow visual inspection of the results and offer consistent results. Kits with glass vial need only 0.5 millimole of racemate. Results are analyzed through optical means and they allow full automation so that library of compounds can be screened at the same time. Types of kits. We have acid kits for base racemate, base kits for acid racemate, and strong acid kits for weakly base racemate. For neutral racemate, it must be brought to either acidic or basic moiety and then you can use the appropriate kits. How do you use the screen kits? You dissolve the racemate in a transfer solvent of your choice and dispense equal amount of the mixture into vials of the 4 kits using multi-channel pipette. For plastic kits, 0.03 millimole of racemate per vial is needed. For glass kits, 0.001 millimole of racemate per vial is needed. Evaporate out the transfer solvent. Add the ChiroSolve solvents provided in a separate plate into the vials. Heat the 4 kits with mixture until the vials have homogeneous solution up to 80 degrees for up to 5 minutes. Cool the kits at room temperature. If needed, freeze them to get the maximum crystals. Pipette out the filtrate and analyze the crystals. Choose the conditions that offer highest quantity of crystals and highest purity. In Ansho prep kits, they include the screen kits. Then they recover up to 90% of racemate back after screening. And then provide components and standard step-by-step procedure for doing the recrystallization steps. They're self-contained. You only need racemate and selector reagents to obtain the final pure enantiomers. It can deliver both positive and negative enantiomer with very high purity. Results are obtained within a week. It defines scalable and consistent chiral separation method. Enantiomer prep kits include screening kits, recovery solution. After screening, contents of the screening kits along with the recovery solution are collected into separatory funnel. Once 2 liquid layers are formed, the bottom layer containing the racemate and solvents is extracted out and dried to recover up to 90% of the racemate. Purification bottles allow easy collection of increasingly purified enantiomer and filtrate during multiple recrystallization. Filtering funnel with vacuum adapter is provided to allow fast crystal separation, and the disposable filters and filter tips eliminate the contamination issues. How do you use the enantiomer prep kits? Using ChiroSolve the screen kits, choose the best chiral separation conditions for the target enantiomer. Collect the contents of the screen kits in separatory funnel and add the content of the recovery solution bottle to this solution. Mix and let stand. When 2 liquid layers form, extract out the bottom layer containing the racemate and solvents and dispose of the top layer containing the resolving agents and solvents. Evaporated solvents from the racemate, weigh the racemate, add equal amount of selected resolving agent and 75 ml of solvent to the racemate and heat the mixture to up to 80 degrees centigrade until it's homogeneous, typically for 10 minutes, and cool it. You may have to freeze it to get maximum results. Extract target enantiomeric salt crystals and store away the filtrate. Add 20 ml of selected solvent to the crystals. Heat and cool until purified crystals are formed. Analyze the crystals. Repeat the steps 7 and 8 until the target purity is achieved. If you need the other enantiomer, extract it from the filtrate and repeat steps 7 and 8 until target purity of that enantiomer is achieved. I hope this presentation was helpful. If you have any questions, please contact us at info@chirosolve.com or refer to our website for further information. Thank you.